Hello everyone, my name is Raissa Krupek and I'm a member of the GVENCK group. And today we are here at the 9th International Meeting on Plant Breeding with Professor James Schnable. Hello, Professor James.
Hello, it's a pleasure to be here.
Thank you. It's a pleasure to have you here as well. Dr. James is a professor in the Department of Agronomy and Horticulture at the University of Nebraska-Lincoln, where his work focuses on maize and sorghum genomics, genetics, and genotype by environment interactions. So, Professor James, we can start our interview with some questions. How do you see the integration of cutting-edge technologies such as RNA sequencing, machine learning, and high-throughput phenotyping in maize and sorghum breeding? And which recent advances do you consider most transformative?
So it's a real challenge as we think about how we integrate a lot of these new technologies. Many of them have been developed in other fields, particularly when we think about machine learning. These are tools that have been developed in areas where data is cheap and there's lots of it available. Particularly in the public sector, we don't have the really big data sets, like if you think about training something for image analysis or language models that are trained on billions and billions of words of text. So I think one of the big things that we have to do as we think about integrating is where do we have strengths in agriculture and where do we need to develop new approaches. So I'm really excited about using things like transcriptomic data, language models that can look at promoters and try and interpret from non-coding sequence how a gene will be expressed. But at the same time, we need to think how are we going to train these models. For promoters, any given species probably has only 20, 30, 40,000 promoters to work with. We don't have tens of millions of records. So getting to the point where we can actually take this and provide information that is relevant to breeders, okay, this is an allele you want to integrate, or maybe even here's an edit we can make to make this gene work better. There's a lot still to be done.
Okay. And what challenges have you faced applying transcriptomics and genomics in field environments, especially in crops like maize and sorghum, which have complex genomes and significant genotype environment interactions?
So I'm going to focus a little bit on transcriptomics, because my guess is you have a lot of people here who can talk to you about using genomics in breeding.
Okay.
Fewer with transcriptomics. The big challenge with transcriptomic data is we can't go back and sample more individuals in the same environment at the same developmental stage. If I'm trying to genotype 1,000 varieties of corn to train a genomic prediction model to conduct GWAS, and then later on I decide, okay, 1,000 wasn't enough, I need 1,500, I can just go, I can get more DNA from more individuals and build up the data set that way. But we can never go back to that same field on the same day for transcriptomics. So we have to plan from the beginning to get data from a large enough population in a short enough period of time that we'll be able to have the power to do the types of analyses we want to do. That's one of the big innovations that my lab has invested in and succeeded at over the last 5 years, is figuring out how can we go to a field and very rapidly collect and freeze the tissue from 1,000 or more kinds of corn or sorghum in less than 2 hours. Because remember, it's not just day to day, it's not just field to field. Even throughout the day, the expression of every gene, almost every gene in the genome, is going to vary according to circadian and diurnal cycles. So we need those really narrow windows. If we can do that, then we get these really powerful data sets that can tell us a lot about what individual genes in the genome are doing in ways that we couldn't figure out using only genomic data.
Awesome. You have founded 3 startups focused on bioinformatics, quantitative genetics, and digital agriculture technologies. How have these entrepreneurial experiences influenced your academic approach and vice versa?
Yeah. So let's start with how the entrepreneurial experiences have influenced my academic work. One of the things that I think entrepreneurial work really helps with is focusing on what matters and what doesn't, and this realization that in many cases we just need to figure out what are the rate limiting steps and solve those. So in many of the projects in my academic lab, I try and teach my students from the beginning it's okay to try a lot of things and then realize we can stop a lot of them. We don't have to finish every project that we start. We need to do just enough to realize, is this something that's going to pay off or not? That's something I think I get from the private sector work. And by trying lots of things and only pushing a few forward, we're going to end up working on much more powerful projects. It's like plant breeding. If you start with 10,000 varieties and you down select to 3 or 4, you'll end up with much better ones than if you just say, these were the 5 varieties of corn I have to make those work. And I think often in academia we fall into that trap of feeling everything we start we have to finish.
Okay. You led the team that sequenced the complete maize genome from telomere to telomere. What were the main challenges and discoveries of this project, and how do they impact our understanding of maize genetics?
So I should actually have a correction here. So I participated in the team. This project was actually led by a really talented professor from China Agricultural University, Zhenxing Lai. And so he invited me to participate as part of the project for this first telomere to telomere maize genome. This was a really exciting project. The first version of the maize genome came out in 2009, which is a lot longer ago than it was, but it still had lots of gaps. There are lots of parts of the maize genome that are very repetitive, and so when you're trying to assemble the genome, it's really hard to put those pieces together. With a combination of new, much longer read sequencing technology and some really patient and determined students and postdocs, Professor Lai and his lab were able to put together this telomere to telomere genome with no remaining gaps. One of the things that is really exciting about that, and one of the parts that I contributed to the most on the project, is this allows us to study a type of genetic variation in maize that was very hard to study previously, looking at what are called nearly identical paralogs. So this is a case where you might have 2 copies of a gene, one after another in the genome. The sequence is almost identical, 99.9% identical. And so almost any previous genome assembly tool would have collapsed those 2 copies together. By having this complete genome assembly that resolves even these duplicates, we're able to identify that in some cases where we think there was 1 gene in the maize genome, there are actually 2. And that maybe that those 2 simply produce more expression, or there may be very subtle functional differences, even though the sequences are so similar.
Okay. How do you see the role of phenomics and quantitative genomics in predicting agronomic traits in maize and sorghum? Are there examples of success in applying this approach to improve productivity and stress resistance?
I don't know that there are really good examples of success yet. I think that there is a lot of potential. If we look at how breeders have developed new varieties of crops like corn over the years, almost all of it has been driven by selection for yield in the target environment. That works really well. But we have a problem, which is that it takes 7 to 10 years to develop a new variety of corn or sorghum. For some crops, like we heard yesterday, sweet potato can be 14 years. And so when you're doing those early stage selections, you're selecting the environment you have at that time. And if the climate is changing, if agronomic practices are changing, if access to fertilizer is changing, the environment in which that crop will be released and have to thrive in farmers' field is farther and farther from the environment where we're doing selections. So historically, breeders could do selection in the current environment, they could figure out what would work well, and then in 7 or 10 or 14 years, it would still do well when it came out into the world. What we want to do with phenomics is start to identify what are the other properties of a plant that determine whether it's going to do well or poorly in different environments. And by understanding that, we can look at our models for what the world is going to look like in 15 years and be developing varieties not for what will perform well today, but for what will perform well then. And that may be different leaf architectures, different photosynthetic parameters, different root architectures, many different things that are going to be adaptive in some environments, detrimental in others, and we can measure those directly instead of only looking at the yield outcome using phenomics.
Awesome, Professor James. In collaboration with other institutions, you have explored the use of sensors and digital technologies to monitor plant growth and health. What are the main advantages and limitations of these technologies in the context of crop improvement?
Okay. So probably the best example I can talk about here, and it's one where I do have a financial conflict of interest because it turned into one of the companies I helped start, is a nitrogen sensor developed with a collaborator of mine, Liang Dong, who was then a professor at Iowa State University. He's since moved to Georgia. So he developed a sensor we could stick into the stalk of a corn plant, and we could measure the amount of nitrate in the actual sap of the corn plant. Now this turned out to have a lot of advantages. If you think about the cost for a farmer of producing a crop, at least in the US, the most expensive variable cost of production is buying the seed itself. The second biggest cost is nitrogen, and it's a huge expense for farmers. It's very energy intensive. It also has a lot of negative environmental impacts. If we apply too much fertilizer, it runs off into the waterways. Lots of bad things can happen. And the most frustrating part is that we know that in about a third of all the acres in the US where corn is grown, and my guess is in Brazil it is similar, the optimal amount of nitrogen fertilizer to apply is 0. But it changes from year to year. Which fields it will be 0, which fields require a lot of fertilizer. So farmers do the optimal thing, which is they apply fertilizer to all their fields, because they'll lose a lot of money if they don't fertilize where they should, and they'll lose a little money if they fertilize where they shouldn't. By being able to measure the amount of nitrate coming up in the stalk of the plant rather than the soil, it turned out we could do a much better job of figuring out during the growing season which fields were going to be nitrogen limited and which ones weren't. So we could give farmers better information about which fields did require fertilizer and which ones didn't. And that's really a triple win. It's a win for the farmer, they make more money. It's a win for the environment generally because we're spending less energy producing fertilizer. It's also a win for the people who live around the farm and can have higher quality water, fewer runoff related events. But that's all production. You asked me about breeding. So now that we can measure this property, how much nitrogen is being taken up, is available in the stalk, this allows us to start asking questions about the functional properties of roots without having to go and dig up roots in the field. First of all, I think often we don't get very good data from digging up roots. The root structure of a corn plant is really wide, and if we dig up, we usually dig up only about this much. But another problem with studying roots through what we call shovelomics, going and digging in the field, is it's really hot, it's in the middle of summer. I spent 2 years trying to study roots that way, and I think I convinced a lot of students they didn't want to be plant geneticists. So now we have a way where we're getting better data on the functional property of the roots by being able to grow lots of corn plants in the same environment and look at how the roots are working. And I can do it in a way where I think I still keep young people excited about going into plant biology, which is a big part of my job as a professor.
Interesting, very interesting. And considering the impact of climate change in agriculture, how do you believe that integrating omics and digital technologies can help develop more resilient crops adapted to diverse environmental conditions?
Yeah. So one answer goes back to what we talked about a little bit earlier, which is that if we can understand how different properties of plant determine genotype by environment interactions and which ones will do well in which environments, we can start breeding and developing varieties for the environments we know are coming, instead of only for the environments that exist today. The other really exciting thing that we can do with a lot of these technologies is we can look at crops that are not the major 4 or 5 big crops that are growing in the world today. So if you think about 1,000 years ago, there were hundreds, if not thousands, of crops that were grown at relatively similar levels of production. Today, a small number of crops like rice, wheat, soybean, maize, are grown on a large proportion of the land and are responsible for a large proportion of all the calories we eat. The reason for that is we've invested a lot in breeding. We've been able to make these crops much more productive, much more efficient, much more resilient. These are all very good things. But thinking about the future and needing to be resilient in a world with much less predictable climates, if we could grow a wider range of crops, that would be good. Farmers don't want to do that today because the yield of these other crops is quite low. Often the agronomic properties are not as good. But we can take the lessons we've learned in taking a crop like corn, where we've increased the yield something like 6x in the last 80 years, and we can take those same genomic breeding approaches, apply them to other crops. For example, I've used this in another one of my companies. We're working with a crop called proso millet that had had flat yields for decades. We took breeding approaches from corn, genomic prediction, statistical field design, and we were able to increase the yield of proso millet by anywhere from 20 to 40% in a breeding program of about 7 years. So think about applying that to a wide range of other crops so that farmers would have a more diverse set of crops that they could grow and still make money, and it would allow us to be more resilient in the face of unpredictable future climates.
Oh, wow. Okay. And in our last questions, what advice would you give to young researchers who want to work with omic in plants, particularly in major crops such as maize and sorghum?
Yeah. I can think of at least 2 pieces of advice. Maybe I'll think of a third while I'm talking.
Okay.
So the first one is that there's a lot of danger in working with really small data sets and then trying to apply all of the really advanced algorithms to those. You can find really complicated ways to do simple things. It doesn't teach you a lot about what you want to learn to be a really good plant geneticist or genomicist. So it's good to seek out really large data sets. Working with the large data sets is how you really learn what works, when it doesn't work, and also how to identify when your data is wrong. That's one of the big challenges we have as biologists, and I think it's one of the things we don't learn just going to statistics and data science courses where they assume you start with good data. So find big data sets. They don't have to be your own. They can be things that you find online and you can apply to new problems, but work with data sets that are big enough to justify the approaches you want to work with. The second piece of advice that I would give, and this has changed a lot just in the last few years, so ChatGPT came out I think about 3 years ago. At the time I was really frustrated. I always make my students learn Python and R, and it felt like they stopped learning it because now they could just ask ChatGPT, okay, give me some code to do this. And that works really well early on because they can do more faster, but then they're not learning enough that once they get beyond what ChatGPT can do that they actually understand, how do I troubleshoot this, how do I write my own script. But in the last few months, we're now seeing sort of command line, agentic coding interfaces that are so good that I think I was wrong. I shouldn't have been pushing my students to keep learning programming. You need to know enough to understand the syntax, but really get comfortable using command line tools in programming and running bioinformatic analyses. I think that if you are able to do this, you'll be able to accomplish a lot more in a lot less time, and you're focusing on the things that humans are really good at. Knowing what are the right questions to ask, and then interrogating when you get a result back, does this actually mean what I think it means? Is the analysis working the way that I think it is? That's also, if you want to be a professor someday, that's a lot of what we have to do, because we're not running the analyses anymore. We're asking some student, analyze this data, and at the end we're saying, okay, you got this result. Is this result what you think it is? Did everything work? And those are the same skills that you use when you're working now with an AI tool that's run the analysis. So you're really getting a practice in that next career stage if what you want to do is to go and do this research in an academic setting.
Okay, these are great advices. Thank you very much. And well, unfortunately, we are already in the end of our interview. I would like to thank you, Professor James, not just for accepting our invitation to our international meeting, but also for sharing your words with us.
Oh, thank you so much. It's been a lot of fun.
That's great. Thank you very much, and we hope to see you in future editions as well.